Hi students, welcome to HSC Biology and Module 7, Infectious Disease. This is video number 23, and this time we're going to look at antibiotics. In our last video, we introduced the idea of antimicrobials as a particular type of chemotherapy and the fact that the antivirals specifically target viruses. Well, this time we want to look at antibiotics, and antibiotics specifically target bacteria. So once again, we want you to start at that entry level, define the term antibiotic, from there to discuss the effects of antibiotics on bacteria, and then also to discuss the need for ongoing development of antibiotics against bacterial infections. And that's kind of the higher level, and we'll look there specifically at some of the antibiotic resistance that's built up in different strains over time. So firstly, antibiotics are chemicals. They are a type of chemotherapy or pharmaceutical drug that target bacteria without destroying the host. So part of what we want to do here is, again, we want to supplement the system so that we can help our immune system to respond quickly and effectively against invading pathogens. This time we're looking at chemicals that particularly target bacteria. We have Alexander Fleming to thank for this, and this is another example of two really key concepts that have often accompanied scientific discovery. The first is serendipity, which is a discovery which wasn't necessarily the original intention that was made by the discoverer. So Fleming didn't set out to discover penicillin or an antibiotic or a chemical substance which would work against bacteria. The apocryphal story is that he was just untidy and he left his plates out, didn't clean them up overnight. They got contaminated with a fungus. The fungus grew in the plates, and the areas around the fungus where some of the chemicals had leached out into the agar had inhibited the growth of bacteria in those areas. Now, that's kind of the general story. There's a lot more to that. And we'll have a look at actually the contributions of both Fleming and Florey in class. But the idea is that it wasn't a discovery that was deliberately made. There's also, and we saw this, we've seen this in stories like the discovery of the process for producing ammonia, which was again discovered by Haber but commercialized by Bosch. And in this case, it was Fleming who was the discoverer of the antibiotic penicillin, but it was Florey and Chan who were able to produce sufficient amounts of penicillin for it to actually be useful. And of course, this became particularly important as we were in 1941 in the middle of the Second World War. And obviously a penicillin that would help stop infections for soldiers who were being shot or hit by shrapnel was a particularly important step forward in our understanding of science and medicine. So penicillin, the first antibiotic, was very, very important, and it was a natural one, but there were others that came behind it. From our natural antibiotic penicillin, which was produced from a fungus, we have been able to synthesize a range of different antibiotics, both partly based on the structure of penicillin and then, I guess, understanding more as we have about bacteria particularly. We've been able to develop different classes of antibiotics, tetracyclines, sulfonamides, erythromycin, and so many more that have continued to develop and actually need to continue to develop for reasons that we'll see at the end of this video. Antibiotics also come in two, I guess, categories. Broad spectrum ones, so the sulfonamides are the broad spectrum ones. So often if we suspect an infection but can't identify the source, a broad acting antibiotic will kill a range of bacteria. Now that's going to kill good bacteria in your body as well as the ones that you don't want. Whereas something that's a little bit more narrow spectrum, like penicillin was, is going to specifically target certain types of bacteria and not necessarily kill off every bacterium that exists. So these are kind of the general types of antibiotics that have been developed since that first discovery of Alexander Fleming's. How do they work? Well, there's three main ways, I guess, that we can discuss the function of the antibiotic. Remember, we're talking chemotherapies, we're talking chemicals. So what do these chemicals do in terms of inhibiting the growth of bacteria? So one thing they can do is they can accumulate in the cells and prevent the formation of cell walls during cell division. So we know in the last stages, when the cells have got all of the information that they need, we have the process of cytokinesis, where the cells are going to split into two. And cells that have a cell wall, the cell wall has to form obviously around the cells at that point in time to completely enclose these two new cells. Now, one of the functions of certain types of antibiotics, such as penicillin, is to build up certain chemical levels that are going to inhibit that formation of cell walls. Another thing they can do, the amphotericins are able to directly destroy the cell membrane. So, of course, if you are able to puncture, otherwise destroy the integrity of the cell membrane, then obviously that's not going to be able to keep the contents of the cell inside. That will lead to the death of the cell. And another thing that antibiotics can do is they can actually interfere with the process of protein synthesis or gene expression. So we know that genes are there in the genetic material to code for the production of certain types of proteins. Those are going to happen through the processes of transcription and translation. And of course, both of these require enzymes to participate in those processes. So if the enzymes are missing, if there's some way you can block the expression of those or inhibit the function of those proteins, then you're not going to be able to produce the proteins associated with those, and that's going to obviously also lead to the ultimate death of the cell. Now, one of the problems that we've had in the fight against antibiotics is that the bacteria colonies, so not the bacteria themselves, but bacterial colonies and populations are building up a resistance to antibiotics. In general terms, if we have multiple bacteria in our bodies, which we do, and we take a course of antibiotics because there's a particular type of bacteria that are causing a problem in our body, what will happen, particularly if we use something like a broad spectrum antibiotic, is going to kill all of the non-resistant bacteria. So anything that's not resistant to that particular chemical is going to die. Anything that is will survive. Of course, one of the problems that we know about bacteria is that they have very fast reproductive rates, so they will very quickly multiply. The other thing with bacteria is they spread very easily and very rapidly from one host to another. So what can happen is that resistant bacteria in your body could just remain in your body and make it very difficult for you if you are prescribed a course of antibiotics at some point in the future. But you could also pass those resistant bacteria on to someone else in your life. They may get sick from that, and then the antibiotics that perhaps did originally work with you to help you to get better won't work with them because you've passed on that higher concentration of the resistant bacterial strains. Probably the best known example of this is the methicillin-resistant Staphylococcus aureus. Sometimes this goes by the acronym MRSA. Now, this is a widespread opportunistic pathogen, so that's the Staph aureus is the pathogen, it's the bacteria, and can affect skin or wound infections, but can also cause pneumonia and septicemia. Now, this MRSA was a particular problem in hospitals for a little while. There were sort of cross-contamination infections that were happening during operations that were happening in hospitals. And so that was a sort of a hospital-acquired MRSA where people were actually getting this secondary infection as a result of procedures in hospitals. But we now understand that there are community-associated transmissions of the MRSA as well, to the point where about a third of the population carries the Staphylococcus aureus bacteria in just your normal nasal microbiota. And about 6% of these are the methicillin-resistant variety. So you have a range, the same thing with E. coli. If E. coli gets into very high numbers, they can make you sick, but there are always E. coli present, and as long as the numbers are sufficiently low and controlled, then generally speaking, they don't tend to do you any harm. Same thing, this particular type of bacteria does live in your nasal passages. It's okay in small quantities, but if the numbers start to rise, they can cause a problem. And also, if they're of this resistant variety and you pass them on to someone who then develops an infection, that may mean that any of these antibiotics are not effective. So antibiotic resistance has parallel development of antibiotics, and this continues to drive pharmaceutical industries to continue to find more and more effective and better antibiotics to continue this fight against bacterial infection. We'll have a look at antibiotic resistance in a little bit more detail in an activity that we'll look at in class. Thanks for watching.